So let's start with the little bit of introduction about biochemistry, and then we go more advanced topic in upcoming days. So in this we will go basically understand how your various complex molecules that are found in biological material, how they work, how they interact within the cell in order to form tissues and then whole organism. So what is biochemistry actually? So biochemistry is the application of chemistry to study the biological processes at the cellular and the molecular level. So in this we are studying the chemistry of um, biological processes at both molecular level as well as the cellular level. And it is emerged as a distinct discipline around the beginning of 20th century when scientists combined chemistry, physiology and biology to investigate the chemistry of living system. So it's a very different discipline beginning of the 20th century when scientists combined chemistry, physiology and biology to investigate the chemistry of the living system. Then principles of biochemistry, it includes cells are highly organized and constant source of energy and is required to maintain the ordered state. Living processes contains thousands of chemical pathways. Precise regulation and integration of these pathways are required to maintain the life. So certain important pathways, example, glycolysis is found in almost all organisms, and all organisms use the same type of molecules, example, carbohydrates, proteins, lipids and nucleic acids. And instructions for growth, reproduction and developments for each organism is encoded in their DNA. So all in all, we can see that this biochemistry, within the cell, they are highly organized and constant source of energy, yeah, and they maintain the ordered state, like everything is in ordered. And several thousands chemical pathways are working, like glycolysis, gluconeogenesis, so many pathways, and they're working very precisely. And integration of these pathways are required to maintain the life. So certain important pathway, example, glycolysis is found in almost all organisms. So all organisms use the same type of molecules, example, carbohydrates, proteins, lipids and nucleic acids. Then instructions for growth, reproduction and developments for each organism is encoded in their DNA. Come on, either calls me. ਇਹਨਾਂ ਦਾ ਹੱਥ ਟੁੱਟਿਆ ਸੀ ਕਿਉਂ what happened so cells, they are the basic building blocks of life, smallest living unit of one organism, grow, reproduce, use energy, they adapt and respond to their environment. Many cannot be seen with the naked eye. A cell may be an entire organism, or it may be one of the billions of cells that make up the organism. And there are basic types of cells such as eukaryotic cell and prokaryotic cell. The prokaryotic cells have the cell wall and the eukaryotic cells does not have the cell wall. So they also have prokaryotes, flagella, spillae, and outer membrane, cell wall, and it has a nucleoid. All the DNA is present in the nucleoid, whereas in the prokaryotes is in the um, nucleus. That's the basic structure. The cytoplasm, microtubules in eukaryotes, lysosomes, SCR, smooth endoplasmic reticulum, rough endoplasmic reticulum, centrioles, yeah, Golgi complex, nucleus, nucleolus, chromatin, plasma membrane is there, rough endoplasmic reticulum, ribosomes, mitochondria, and so on. So they contains three basic cell structures, that is nucleus, cell membrane, and cytoplasm with organelles. So nuclear region contains the DNA, the cell membrane, and the cell wall, and they contains ribosomes, no membrane to make proteins in their cytoplasm. So they also have these flagella pillae to move around, and they have this nucleoid region in these prokaryotes. So that's the main difference between your eukaryotes and the prokaryotes. Then water is very essential in our body. If somebody is interested to study about proteins, so they also have to consider water is in our body is made up of 70%. Our body has 70% water. So considering that, so all the protein structures, they have these these water molecules bonded to the whole protein molecule. So that's why we say that our body is around 60 to 90% is of water, and it's used in most of the reactions. So it's very, very essential, very, very important. Yeah. And it's also called as universal solvent. And it's also called as universal solvent. So biomolecules, so just like cells are building blocks of tissues, so likewise molecules are also building blocks of cell. So animal and plant cells contain approximately 10,000 kinds of molecules. So water constitute around 50 to 95% of cells content by weight. Ions like sodium ion, calcium, potassium ion, calcium ions may account for one another 100%. So almost all other kinds of biomolecules are organic, that is see carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. So all are very important. And there are infinite variety of molecules which are containing your carbon. So basically carbon is playing a big role from many perspective. And most biomolecules considered to be derived from the hydrocarbons. And the chemical properties of these organic biomolecules are determined by their functional groups. So most biomolecules have more than one. In order to understand this, let me take you Gmail. No. So here I will be discussing various sizes. You have a coffee bean, 12 multiplied by 8 millimeter, and times regular point is 12 point. and there's a grain of rice with 8 by 2.5 millimeter, and sesame seed is around 3 by 2 millimeter, and grain salt is around 0.5 millimeter. And amoeba, let's go to the now prokaryotes. Now you can see their size is around 500 micrometer, paramecium 210 to 60, and then we go for human egg 130 micrometer. The photoreceptor that are present in our eyes, rod eye cells, 2.5 micrometer. And sperm 60 by 5 micrometer, skin cells 30 micrometer, RBCs 8 micrometer, and your chromosomes 7 micrometer, yeast 3 micrometer. Now this was and your E. coli bacteria. So this was all about your chromosome, RBCs. Yeah, so they're all in the micro range. Mitochondria also 4, E. coli 3, and your lysosome is also 1 micrometer. Now let's talk about viruses. Measles virus 220 nanometer, influenza virus is around 130 nanometer, and HIV virus is around 130 nanometer, and hepatitis to be 45 nanometer. Yeah, and the rhinovirus is around 30 nanometer. Coated vesicles 90 nanometer, then ribosomes 30 nanometer, hemoglobin 6.5 nanometer, yeah, antibodies 12 nanometer, tRNA 7 nanometer, and phospholipids 0.9 multiplied by 3.4 nanometer. And there is adenine, that is the nucleotide. Teras, it is 1300 by 760 picometer. Now things are coming into the picometer, that is 10 to the power 12 minus 12. Your water molecule H2O 275, methionine which is an amino acid 1100 picometer, and then carbon atom just size of 340 picometer. So this will be very interesting, as we have just discussed the biomolecules, right? And how biomolecules are very essential and their various characteristics. So it will be very essential to know their sizes also, how each size matters a lot. And yeah, so starting from carbon, which is essential part, with which all your picometer, nanometer, micrometer, then millimeter, all are interlinked with each other. So this slide is very essential to understand that how everything is co-related with each other. I hope we have not done this part before. Yes, we have not done this part. I got one assignment last night, I think. It was Sharvari Pujare. That's very nice. Thank you. So you made a very nice like summary that I can see that you have understood it by yourself while you are making that, and that's it. That's all I want, that you're able to make sense that what we are talking, and you know what is, and enzyme present in sequence, topoisomerase, yeah, tools used in this, PCR, DNA isolation, yeah, that's it. So I mean there is a detail about it that you can read through the presentations, but moreover I really like it, like this should be a nice summary of everything. Appreciate that. Yeah, then there is a thing called Catabolism and anabolism. So your catabolism is breaking your large group into the small group, and then during that time energy is released. But making from small group small building blocks to big macromolecules, then ATP energy is required, and that is anabolism. And together, this catabolism and anabolism is known as your metabolism. Together, the functions together in like yin yang pattern. In the Chinese culture, there is a yin yang. So both are interconnected with each other, and they cannot live without each other. Yeah. So that's how this building block and macromolecule works. So building blocks, for example, simple sugar, the macromolecule would be the polysaccharides. And for the amino acids, there will be protein. For nucleotide, there will be RNA or DNA. And for the fatty acids, there will be the lipids. So from macromolecule to the building blocks, if they are being made, like breaking is happening, then catabolic. If it is from building block to macromolecule, then it is anabolic. Then there is a thing called linking monomers. So two sugar group could be attached to each other by removal of the water. If we can see there is OH present, OH present. So from here maybe we can remove OH, from here H, then H2O will be made, and they will be all together joined by the oxygen bond. Yeah. So cell links monomers by the process of dehydration synthesis. So removing a molecule of the water. So this process joins two sugar monomers to make a double sugar. So from here H could be removed, from here OH could be removed. So by removing OH from one side and moving H from the another side, you have H2O forms, and hence they could be joined together. What if they are joined together by the same bond oxygen? They could be also broken in the same way. So the polymers within the cell, your macromolecules, could be break down by the hydrolysis. So in this is a dehydration synthesis, that is we are removing the water. In the hydrolysis, that means we are adding a water molecule. So the same way in the previous reaction, we have this two monomers joined together by oxygen, and then by the addition of water, so twos are separated. So they all got their water, OH, OH, and then they are separated. So both reactions are interconnected with each other. So water is added to split a double sugar. Then there is a sugar, which is the most abundant organic molecule that is found in nature, and initially synthesized in plants from complex series of reactions involving the photosynthesis. and they are the basic unit is your monosaccharides. From monosaccharides you have your disaccharides, trisaccharides, yeah, and many more sugars are possible. So main functions of these sugars is to store energy in the form of starch, yeah. So photosynthesis in plants or glycogen in animals and humans. So in the plants they are stored as a starch, and in the humans they are stored as a glycogen, and they provide energy through metabolism pathway and cycles. Like your glycolysis, Krebs cycle, TCA cycle. So I think we will do all of them, and they supply carbon for synthesis of other compounds. And they also form structural components in various cells and tissues. So they are very important part of the metabolism of the cell. And they also helps in to communicate between the two cells. So they are also required for the same for intracellular communication. Now we have discussed so far the sugars. We have discussed about how linking and breakdown of polymers is happening, monomers and polymers. We have discussed about the biomolecule structures, what are the various biomolecule structures looks like. We have discussed some very nice characteristics of biomolecules and how they are important in our body so far. And we have discussed that water, which is an essential part of a body, is also called as universal solvent. So all things that we are discussing here are we are discussing from the perspective of biochemistry, yeah. And we have made this point in the starting of the lecture that there are two different cell units, that is eukaryotic and prokaryotic, and they both functions according to their types. Yeah, some principles behind the biochemistry. Then we have discussed the definition of biochemistry, some objective. So I will keep in the lectures going on. While we are done with the 50% of the topic, I go back in order to have in touch that what we are going through, so that we do not lose the connection. So everything is in the flow. Yeah. So this is what we have discussed so far. Sugars. Now we'll take to another biomolecule, that is fatty lipids, fatty acids. So they are made up of polar head, which has hydrophilic end, and non-polar tail, which has hydrophobic end. So this is the butter in which your fatty acids are present. So monocarboxylic acids which contain even number of carbon atoms. Uh, So there are two types. One are saturated, one are unsaturated. The saturated are the one with the single bond, and the unsaturated are the one with the double bond. So fatty acids are components of several lipid molecules. So example, lipids are triacylglycerol, steroids, for example, cholesterol, sex hormones, and then there's fat soluble vitamins also. And the function, as in the fatty acids, is to store the energy in the form of fat. They are membrane structures, and they act as an insulation, thermal blanket to all the organelles or to the cells or to the organs in our body. And they are also reason for the production of hormones. Synthesis of hormones is possible. So there is steroids. So the carbon skeleton of steroids is bent to form four fused rings. That is cholesterol. Um, Cholesterol is actually a base steroids which our body produces, and there's other steroids our body is producing, that is estrogens and testosterone. Yeah, testosterones are produced in the males mostly, and estrogens are being produced in the females. And because of that, we have this male characteristics. The more testosterone we have, the more will be the building muscles is helpful in it. And estrogen is from the female perspective for the yeah, for the reproductive system, for yeah, what is called menstruation cycle. So for all these, this estrogen hormones is very important. So all these steroids, if their volume in our body gets less, could be given out from outside in artificial manner in order to maintain the proper equilibrium. Yeah. So here you have to consider the structure. So there is a uh, six six membered ring, and there is a five pentagon structure. Then here we have hydroxyl group OH, methyl group. On the top there is a chain of alkyl groups. So 1, 2, 3, 4, 5, 6, 7, 8. 8 carbon chain is there on the top. Whereas in the testosterone, you only have hydroxyl group present here and double bond of oxygen. And the estrogen is the same structure, is just you have on the top, instead of hydroxyl group, you have O or double O bond here and OH bond here. So they got interchange with each other. Yeah, so that's the main difference between your testosterone and estrogens. They just oxygen bond and the hydroxyl. They just interchange the space, and you can see there's a drastic effect of these both hormones. Sometimes in the bodybuilding world, the female contendants, they tend to, in order to build muscles, we need more testosterone. Yeah, because as a female, from the natural perspective, from the back in nature, they tend to produce more estrogens. Yeah, but not testosterone. But in order to build muscles, they need to take this testosterone from the external perspective in order to build the muscles. So for that, when they're taking these testosterones, so what will happen, their voice will be also male because of that. Yeah, there will be some genetical changes at the DNA level because of giving a body something which is not natural. If your body, you're giving more male hormones, so it will start it to act as a male also. And the same way, if the male one, um, they tend to eat more estrogen products. So let's say they're saying soya bean now. So it's a myth, I don't know what it is, but they say for the vegetarian soya bean, if somebody is eating a lot, then they might have estrogen hormones to be more produced in them. And then which will leads to have a male breast, and like many other perspective which will be from the female perspective in the male. So remember, whenever yeah, if we get a chance in the laboratory to get our test done, get your testosterone or estrogen level checked, and whether they are in the right order, because we don't tend to check these hormones level in our body. We just, like, in my childhood, I never, because it's, we are growing naturally, but if some hormones or some medication is going on, like somebody has thyroid problem, we will discuss about that. There is thyroid stimulating hormone, there is a T3 hormone, there is a T4 hormone. Yeah, so because of these hormones, so some changes could happen to the testosterone and estrogen level also. So everything is dependent with each other. So after three, I mean, you're all young, so don't worry, but later on, by the 40s and 30s, things start to change. Body is not working as it should be. So synthetic anabolic steroids are variants of testosterones. Now comes your nucleic acids, which store hereditary information, which contains information for making all the body proteins. There are two types, as we know, DNA and RNA. Yeah. This we have discussed in very well manner in the last weeks of lectures, I guess. Um, so I think it's not required to repeat anymore. Then comes your amino acids. So as we know, amino acids, they have alkyl group, that is alpha carbon group, attached to the alpha hydrogen group in the center. And to the left, your alpha amino group is there, and to the right, you have your carboxyl group, COO, right? Your NH3 gives a positive charge, whereas COO gives a negative charge to the amino acid. Where on the top, your alkyl group, um, it has a side chain. This is giving a, yeah, according to that R group, the alkyl group is attached there. So the basic properties of amino acids are changed because of that only. Whether your amino acid will be polar, unpolar, positive, negative, and so on. So building blocks of proteins. Yeah. So building blocks of proteins. So their alkyl group side chains determines the chemical properties of each amino acids. As we have discussed, also determines how the protein folds and its biological function, and functions as a transport protein, structural proteins, enzymes, antibodies, and cell receptor. So proteins as enzymes. So as we know, we will discuss about enzyme in totally complete batch, how the mechanism of enzymes work, what are the basic types of enzymes are, how they affect the activation energy. We will discuss the kinetics, inhibitors later in detail. But many proteins act as a biological catalyst or enzymes. Uh, so thousands of different enzymes exist in the body, and they control the rate of chemical reactions by weakening bonds, thus lowering the amount of activation energy needed for the reaction. So they are also known as catalyst. So no, not interfere with the equilibrium of reaction, and they could be reusable. So we can say these enzymes has a very specific function. They are very specific in nature. Yeah, they could be reusable as much as they want, as they could do. And what they do is that there is a need, in order to make the reaction done, there is a need of, initiation reaction, like to make your reaction start, and that reaction is known as the activation energy. And these enzymes basically helps to lower that activation energy. Yeah, and also they tend to make the reaction very fast. So whatever the product need to be produced, they will be produced really fast in the case of enzymes. And yeah, there's a very less byproducts are present, very less toxic, no inflammables are produced. But on the other side, they are a bit expensive, of course, time consuming, and yeah, so that's the basics cons, but there are lots of pros of using enzymes, proteins as enzymes. So these enzymes has a cleft or groove in which your substrate or fraction reacts, where your substrate is attached to your enzyme, and that step is known as your active site. So nature and arrangement of amino acids in the active sites make it specific for only one type of substrate, which accept just one enantiomer. Yeah. So in all, so what we have discussed is that what are the basics of biochemistries are. We have discussed various biomolecules of the life, and we have discussed also types and functions of these biomolecules. Yeah. I hope I try to do this slow, because the more we go in detailed manner, so the more it will be difficult to understand then. So I will share these lectures. Okay. So बच्चे, I think um, we are I if I start my next topic, it will take me another 20 minutes or 30 minutes, and it will be, yeah, will not make sense to in between stop it. So if you have any questions, please let me know, um, and then we will wrap it for today. Okay, students, so that's it for today. We see us tomorrow, same time, with about to talk about amino acids and lipids. So we'll discuss about amino acids tomorrow in details, and yeah, further different things. So thank you very much. Wish you a nice day. Take care. Bye bye.